Bladder EpiCheck DNA extraction. Materials required but not provided. 20, 200, 1000 microliter filtered pipette tips. 20, 200, 1000 microliter adjustable pipettes. Ethanol, 96 to 100%. Do not use denatured alcohol which contains other substances. Microcentrifuge with rotor for 1.5 milliliter tubes able to reach at least 12,000 Gs, not a benchtop mini centrifuge. Vortex. For the first use of DNA extraction kit, prepare the following reagents according to instructions: protease, NW1 buffer, and NW2 buffer. Gently mix each reagent before use. Do not vortex. If a precipitate has formed in the buffer NL, dissolve it by incubating at 56 degrees Celsius. Change tips between samples in each step of the protocol. If the cell pellet is frozen, thaw it at room temperature and briefly spin before use. Use the test form to document the date of extraction, the operator, and the lot number of the extraction kit used in this step for future reference. Use the Bladder EpiCheck DNA extraction kit. Mix the protease stock solution and load 20 microliters directly into each sample. Gently mix buffer NL and add 400 microliters to the sample. Vortex immediately for 15 seconds. Do not batch samples for vortexing. Do not add protease directly to NL buffer. To ensure efficient lysis, mix immediately and thoroughly for 15 seconds. Incubate at room temperature for 10 minutes. Briefly spin down the sample to remove drops from inside the lid. Add 400 microliters of ethanol to the sample. Mix again by vortexing. Briefly centrifuge the sample to remove drops from inside the lid. Take the appropriate number of mini spin columns. Load 700 microliters of the lysate to the mini spin column. Do not load pellet or any viscous parts to avoid clogging the membrane. Do not discard the rest of the sample as it will be used in the next step. Centrifuge at 6,000 Gs for 1 minute. Place the mini spin column in a clean 2 milliliter collection tube supplied in the extraction kit and discard the tube containing the filtrate. Apply the remaining lysate, 500 microliters, to the mini spin column. Do not load pellet or any viscous parts to avoid clogging the membrane. Centrifuge at 6,000 Gs for 1 minute. Place the mini spin column in a clean 2 milliliter collection tube and discard the tube containing the filtrate. Mix buffer NW1. Load 500 microliter buffer NW1 to the mini spin column. Close the cap and centrifuge at 6,000 Gs for 1 minute. Place the mini spin column in a clean 2 milliliter collection tube and discard the tube containing the filtrate. Mix buffer NW2. Load 500 microliter buffer NW2 to the mini spin column. Close the cap and centrifuge at full speeds for 3 minutes. Place the mini spin column in a clean 2 milliliter collection tube and discard the tube containing the filtrate. Centrifuge at full speed for 1 minute in order to eliminate buffer NW2 residuals. Observe the collection tube. Only a tiny drop should be left on the bottom. In case of a significant filtrate volume, place the mini spin column in a clean 2 milliliter collection tube and centrifuge again at full speed for 1 minute. Place the mini spin column in a clean 1.5 milliliter safe lock microcentrifuge tube supplied in the extraction kit, and discard the collection tube containing the filtrate. Mix buffer NE. Open the mini spin column and add 100 microliter buffer of NE. Incubate at room temperature for 1 minute, then centrifuge at 10,000 Gs for 2 minutes. Discard the mini spin column. The filtrate in the 1.5 milliliter safe lock tube contains the eluted DNA. If you do not proceed immediately to DNA digestion, store extracted DNA samples at -20 degrees Celsius. DNA samples can be kept up to 30 days at -20 degrees Celsius. In this video, we reviewed the DNA extraction part of the assay. Please follow the manual for guidelines. Here are a few points to remember. Do not add protease to NL buffer. Add directly to each sample. Vortex for 15 seconds immediately after adding NL buffer. Vortex 1 sample at a time. Do not load pellet or any viscous parts to avoid clogging the membrane. At the residual elimination step, make sure only a tiny drop is left on the bottom of the collection tube. If not, use a new collection tube and centrifuge again.